Welcome to our lecture on Chapter 15 and 16 covering microbial mechanisms of pathogenicity and disease and epidemiology. We'll explore how microbes cause disease, the factors that contribute to their virulence, and how diseases spread within populations. Before we go any further, though, we really need to lay the groundwork with some basic terminology. The first is, what is pathogenicity? Pathogenicity refers to the ability of a microbe to cause disease by overcoming the host's defenses. And disease is any condition in which the normal structure or function of the body is impaired. Virulence, on the other hand, is the degree to which an organism is pathogenic. So, in this chapter, we will analyze the mechanisms that microbes use to establish infection and cause harm. Infectious diseases are caused by pathogenic living organisms invading the body. These diseases can spread through the body through various means, which we will explore later in this lecture. However, diseases can be either infectious or non-infectious. Non-infectious diseases are not the area of focus for this class, since they are caused from genetic and environmental reasons. Rather, we will be focusing on infectious diseases that are commonly the result of microscopic pathogens. So, how do pathogens cause disease? Well, pathogens follow a general sequence to establish infection. First, they enter the host through specific portals of entry. Then they invade and infect host tissues or host cells using certain virulence factors. And third, they have to exit the host in order to infect another host. So they can exit via specific portals to continue the infection cycle. I want you to imagine you're at a crowded airport, someone sneezes near you, releasing millions of tiny influenza virus particles. One lands on your hand, you later rub your eye, the virus enters through the mucous membrane of your conjunctiva, and within hours it's multiplying inside your body, inside of your cells specifically, preparing for its next move. Well, welcome to the world of microbial invasion. Pathogens enter the host through various mucous membranes, including the respiratory tract or inhalation, gastrointestinal tract or ingestion, genitourinary tract, or the conjunctiva, in other words, the eye. They can also enter through the skin, such as via natural openings, hair follicles, or sweat glands, through direct penetration, such as cuts and wounds. And we might refer to breaches in the skin as the parenteral route. This involves some sort of cut or lesion in the skin or mucous membranes through injuries or medical procedures, and microbes can gain access into our bodies through that mechanism. Some pathogens require a special entry portal to cause disease. For example, Streptococcus pneumoniae causes respiratory disease, but not gastrointestinal disease. And again, E. coli can cause infections in the genitourinary tract, but not usually in the gastrointestinal tract. After entry, microbes must adhere to host cells. They do this using surface molecules called adhesins, which allow them to attach firmly at the portal of entry. Pathogens use virulence factors to evade the host immune system and establish infection. Different types of microbes have different virulence factors or virulence mechanisms. For example, bacteria may use capsules, exoenzymes, and toxins, and we'll talk about those in a little bit. Viruses can use their adhesins or antigenic variation. And eukaryotic pathogens can also take advantage of biofilms, and immune evasion strategies. Let's take a look at some of these in bacteria first. Some bacteria have a rigid glycocalyx known as a capsule, which is tightly organized and attached to the outer surface of their cell envelope. And this can protect them from phagocytosis or being consumed by the immune cells. For example, only encapsulated Streptococcus pneumoniae, so those Streptococcus pneumoniae that have obtained the gene for making a capsule and express that gene, can actually cause pneumonia. So think of, in other words, you might use the analogy of thinking of a bacterium as a medieval knight. And without armor, it's vulnerable to enemy attacks or phagocytosis. But if it wears a capsule, it becomes heavily armored and protected, much like you might think of a knight, and can evade destruction. And this is how Streptococcus pneumoniae is able to survive inside of the body. Other bacteria have a slime layer, and this is a loosely attached glycocalyx that helps them form biofilms. Staphylococcus epidermis, a common bacterium that's found on all of our skin, is actually a common cause of catheter-related infections in hospitals. Infections in hospitals are called nosocomial infections, so I might use those two words interchangeably. And it uses a biofilm for protection against immune responses. Certain bacterial structures enhance virulence as well. For example, there is something called the M protein in a bacterium called Streptococcus pyogenes, and this allows it to resist immune attacks. Streptococcus pyogenes is commonly found in skin lesions. And you can also have the mycolic acids, which we've discussed when we went over staining and smearing. In Mycobacterium, this would be particularly important for Mycobacterium tuberculosis, which provides resistance to phagocytosis, sort of in a similar way that you can think of the glycocalyx doing so. Several bacteria produce something called exoenzymes that aid in invasion. Enzymes, again, are protein catalysts that help speed up reactions. And the prefix exo just means that they release these or they exit the cell. And so they're found outside of the cell. There are many of these that bacterium use in order to assist in their infection. Well, a very common one is called coagulase. And remember, anytime you see a word ending in ase, A-S-E, that tells you right off that that is an enzyme. And coagulase causes blood coagulation or blood clotting. And that clot can build a barrier and protect that bacteria from immune cells being able to reach it and digest that bacterium. Here we have collagenase, and collagenase breaks down collagen. And this allows bacteria to invade new tissues of the body by breaking down that source of connective tissue. Phospholipase C is a enzyme that helps Bacillus anthracis escape immune detection. And what that does specifically is it allows the bacterium to break through the phagosome. So when seen in this image here, you have a immune cell and it goes through phagocytosis. And when it enters inside of the cell, it sends it to something called the phagosome here. And within the phagosome, this is a organelle of that immune cell that has a bunch of enzymes. And what it does is it breaks down that cell and destroys it. And this phospholipase C allows it to break through the lipid membrane and escape being digested. And another common enzyme is that of hyaluronidase. This is secreted by Staphylococcus aureus. Hyaluronic acid, also known as hyaluronin, shown here in this image, This is a naturally occurring component of the extracellular matrix of connective tissues and skin and cartilage and the synovial fluid. And this provides structural support and lubrication and hydration in those tissues. And you can imagine your body, the cells in your body, they're made up of this brick wall. And the mortar between the bricks or your cells is this hyaluronic acid. Bacteria like Staphylococcus aureus produce hyaluronidase, and that enzyme can dissolve that mortar, allowing that bacterium to invade into new tissue spaces and spread like a thief breaking into a building. So, moving right along and talking about toxins, toxins are biological poisons, and those can increase the virulence by allowing those pathogens to invade their host and cause damage. Here are a list of two broad categories of these toxins. One are called endotoxins, and the other are exotoxins. So, in general, every toxin can be put into these two categories. Endotoxins are released when the cells divide or die, and they are found in not outside of, but in the outer membrane. So they're attached to that bacterium. And they're typically associated with gram-negative bacteria that have that outer membrane that you can attach things to on the outside. Whereas exotoxins, these are compounds, they're mostly found in gram-positive bacteria, and they're part of metabolism, and they can be released to produce specific damage to host cells. So again, toxins, they're biological poisons that increase virulence. They can be classified into exotoxins, which are secreted by bacteria to target host cells. And again, that's typically in gram-positive bacterium, and endotoxins are typically components of gram-negative bacterial cell walls. And we'll get into a couple of those here in a second. But here's a list of common exotoxins. I just chose the four here to go over. One is cholera toxin, and that's from Vibrio cholerae, which causes cholera. And this causes the secretion of fluids and electrolytes from cells. And so this is part of the reason why this organism causes dysentery. The next one is tetanus toxin. As you can imagine, there's a theme here. This comes from the organism Clostridium tetani, which causes tetanus. And this inhibits neurotransmitters and can cause, and does cause paralysis. Next is that phospholipase C. It's from Pseudomonas aeruginosa, and this disrupts cell membranes by destroying phospholipids, so it allows it to escape that phagosome. And one that's kind of similar to that, its mechanism is different, of course, is streptolysin. Streptolysin comes from Streptococcus pneumoniae, which can cause pneumonia. And this streptolysin actually disrupts cell membranes again. Rather than attacking phospholipids specifically, it opens up a pore. So it creates a pore channel that these bacterium can use both to allow those digestive enzymes to be leaked into the rest of the cell, that the immune cell that consumed them, but also as a method of passage for it to escape the phagosome. Here we have our endotoxins. The lipid A, so if we look here, this is LPS. We've looked at LPS in the past before. It stands for lipopolysaccharide. Lipo meaning it has that lipid portion, this lipid A. And then the rest of it, the core, the O antigen, all this stuff, you don't need to really memorize, except for you need to know that these are all representative of sugar. So these are kind of like a skeletal structure example of a bunch of these sugars that are linked together to this lipid A. And the lipid A portion, if we look down at this image of a gram-negative bacterium, the lipid A is embedded in the outer membrane. And the polysaccharide portion is what is exposed to the outside. And this lipid A part, it can be toxic to us. And in fact, um, lipid A is released when gram-negative bacteria either die or through the division process, some of those cell wall parts just naturally slough off through the process of binary fission. This part of the LPS, one of the big issues with it is it is not destroyed by heating or cooking. So you can imagine if you have a bunch of gram-negative bacteria and they grow up in, say, a soup or a gravy or something like that. And then you think, well, I'll just microwave this a lot or cook it on the stovetop again, reheat it really hot and kill all of the bacteria. Great, you make it kill all of the bacteria. But if there was a lot of bacteria that grew in there, that lipid A can still cause a disease. It can cause a inflammation and damage to the body. Another situation where this comes up is with antibiotic treatment. When someone is suffering from a large infection by gram-negative bacteria, taking a large dose of antibiotic treatment and immediately zapping those bacteria and killing them off results in the cell wall parts leaking out of those cells. And the lipid A that's present in that LPS of that gram-negative bacteria can potentially lead to something called septic shock. And what happens with septic shock is this is a basically an acute inflammation throughout systemic inflammation, and that leads to a massive drop in blood pressure, and this can result in multi-organ failure and even death if there's a high enough concentration. And there is an LD50 for the amount of lipid A that is lethal. We'll discuss LD50s in a moment, but basically it stands for a lethal dose in 50% of the population. And it's about 0.24 milligrams per kilogram. So if, let's say you have a 150-pound person, that's about 300 kilograms. So that's it's not too important, but it's about 30 grams of lipid A would kill off about 50% of the population. We're not going to bother memorizing this value, but I just left it here to show you it's important to get a little bit more familiar with terms like LD50 and ID50 that we'll discuss later in this lecture. Now that's step one. Now we're going to get to step two, and that's how the pathogen can invade and infect host cells. And viruses, for example, can use adhesins to help facilitate the entrance of host cells. Adhesins, again, these are structures that can bind to host cell surface molecules such as proteins and sugars on the outside of cells. And once they bind to them, this can then help facilitate the next step in the viral cycle, and that is to gain entrance into the cell. So a perfect example of this, and one that we've discussed before, is hemagglutinin, which is present in the influenza virus. And this hemagglutinin, actually shown here, can bind to the sialic acid that we have on the outside of our cells. And so that allows that viral particle to attach firmly to those cells. Our immune systems recognize pathogens by the components found on the outer surface of those pathogenic cells or agents in the case of viruses. And these components, we call those antigens or antigens. Pathogens that change their surface proteins, allowing for antigenic variations, can evade immune responses. This is like an analogy of if you have someone who committed the crime, they change their disguise in the middle of a police chase so that they can evade being caught by the police. This is seen in many pathogens such as Neisseria gonorrhea, which causes gonorrhea, HIV, which causes AIDS, and Giardia lamblia, which causes Giardia. This, however, is especially common in influenza viruses, and this requires new vaccines annually so that our immune systems can be trained on the seasonal antigenic variation. Our immune system is designed to recognize and attack invading microbes and usually does so effectively. But some pathogens, like the ones we discussed, such as Neisseria gonorrhea, can constantly change their outer proteins, making them unrecognizable to immune cells. Therefore, infections can persist even after an initial immune response. The term antigenic variation generally refers to the ability of some pathogens like bacteria or parasites to alter their surface antigens. And this is often through mechanisms like some sort of gene conversion or genetic recombination of their genes that code for those outer surfaces. And that allows them to, again, evade the host immune system. And this variation can lead to significant changes in antigenic structure, allowing the pathogen to repeatedly infect the same host without the host being able to gain long-term immunoprotective effects and train their immune system on those variants. So we can see an example here. Here's this variant 1, and it has these outer cell surface proteins, and we develop antibodies that can recognize some of those. However, there might be another variant that has a different shaped outer surface receptor, and we no longer recognize those, so on and so forth. And so new variants can occur throughout time. The influenza virus changes over time through two main processes: antigenic shift and antigenic drift. They sound similar, but they're monumentally different. These changes mainly affect the two proteins in the virus's surface, hemagglutinin, or HA, and neuraminidase, NA, which are important for the virus to infect hosts and for our immune systems to recognize it. Antigenic drift is like the virus slowly tweaking its look over time. This is because the virus's RNA polymerase isn't very accurate. It makes small mistakes or mutations in the HA and NA genes. As it reproduces and makes copies of its genome. And this happens in influenza viruses, leading to little changes that help the virus dodge our immune system. That's why we get seasonal flu outbreaks and need a new flu shot every year. Antigenic shift is a bigger deal. Picture this: when different strains of the virus infect the same cell, they can swap parts of their genetic material, creating a whole new combination of HA and NA. This can happen when animal and human flu strains mix, or maybe the same human flu strains mix together too. But when a different animal and human flu strain mix together in the same host, this can lead to even bigger issues. This can lead to a virus that's so different, almost no one has immunity to it, which can cause a pandemic. So you can take a look at the H1N1 influenza virus happened in 2009 as an example. And it was born from this kind of shift, mixing genes from swine, bird, and human flu viruses. On the other hand, influenza B viruses don't do this because they mostly stick to humans and don't have a large animal pool to mix with. So while antigenic drift gives us those yearly flu variations, antigenic shift can shake things up big time with influenza A, potentially leading to pandemics by introducing completely new HA and NA types. And this is just kind of repeating what I just said here. And antigenic drift are small genetic changes over time due to just their replication of their genome, whereas the antigenic shift occurs when two different influenza viruses share genetic parts, creating a whole new antigen. Okay, so now let's talk about some fungal virus factors as well. Fungi, as we've discussed in the past, can cause infections. They can do so through producing a biofilm production, and they produce something that we call capsules. They're not the same as bacterial capsules, but it's the same sort of mechanism here that provides them a protective coat. One of those is Cryptococcus that produces a protective capsule that helps it resist phagocytosis. And a third virulence factor are mycotoxins. Now, myco just means fungus. Mycorrhizae. When you ever see the prefix myco there, you should just understand that it's talking about fungi. And two fungi in particular, we have Claviceps purpurea, and this produces the hallucinogen ergot. This is a big issue for us because this particular fungi grows on wheat, and wheat that is contaminated with this particular fungi can produce ergot. And ergot is very similar to what's found in LSD. And so, of course, this can be a bit of an issue. And there's a lot of really interesting, I won't get into it, but historical stories involving ergot causing issues in towns and villages throughout history. Although a lot of it is not really founded in definitive proof because at the time we didn't know about microbiology, so we couldn't go through a proper scientific process of vetting it out and determining it was Claviceps purpurea. So the next one is Aspergillus flavus. And Aspergillus flavus can potentially produce a mycotoxin called aflatoxin. And the problem with aflatoxin is it's a mutagen. And so it can raise the risk of getting cancers and other diseases. And this particular fungi can commonly grow on legumes. Most commonly, we see it issues with peanuts and peanut butter. So if peanut butter is not properly peanuts that go into making peanut butter is not properly examined and tested for aflatoxin, you can have a food recall on peanut butter, and it happens quite a bit actually. All right, so let's talk about protozoan virulence factors here briefly. They can also go through antigenic variation, like I said, Giardia lamblia can, as well as a protozoan called Plasmodium falciparum. This is a very important one for you to be familiar with because it causes this particular protozoan can infect your blood and it causes malaria. And it goes through antigenic variation over time. And this is why just because you got malaria once, it doesn't actually protect you from not getting malaria again. Now, these organisms also have special attachment mechanisms. We discussed this in chapter 5, the eukaryotes of microbiology, but Giardia lamblia, for example, has that adhesive disc. It allows it to attach tightly to the intestinal epithelial cells. And they can create capsules as well. The Trypanosome brucei, which causes African sleeping sickness, produces a capsule, and that protects it again from phagocytosis. The final step is how it exits the host in order to continue the pathogenic cycle. And so pathogens have to leave. They typically leave through specific portals. This could be respiratory, such as coughing and sneezing. It could be gastrointestinal, such as the feces or saliva, or genitourinary secretions or skin or wound drainage. Now, we're going to get into determining the cause of an infectious disease, but before we get there, let's take just a pause here and go over a couple more vocabulary words, and that is signs and symptoms. You've probably often heard of those two put together when doctors are assessing someone and seeing, if they have a disease, they're looking for signs and symptoms. And they're different categories. A sign is a disease sign is something that we can observe as a third party of of a disease. So a healthcare worker can actually measure it. They don't need the patient to tell them about it. It's objective, it's measurable. So you're looking at the vital signs. That is something that you can objectively measure. A symptom is the way the patient is feeling. So it might be very well happening to the patient, but it's not something that you can measure specifically. An example of this could be nausea or fatigue. So nausea, we can't measure it, but they can tell us about it. However, if the patient vomited, then of course that now becomes a sign that we can measure. A patient might say that they feel like they have a fever, and this is where it gets a little bit murky. And the feeling of a fever does not necessarily mean you have an elevated temperature. A healthcare worker can measure an elevated temperature, and now that is a sign, but the sensation of a fever may not necessarily be a sign, but rather a symptom. But there are parts to a fever that could be measurable. And so in that example, you can see that sometimes the two can contradict each other. Sometimes a patient might feel like they have a fever, and while that sensation might be very real for them, there might not be a measure of elevated temperature, for example. All right. So, Robert Koch was the person who first started connecting disease to microbial agents. And he developed something called the Koch's postulates that were used in order to directly determine the cause of a disease as being from a infectious source, from a living source or a viral source. And so there are four postulates that you have to go through. First, the suspected pathogen, it has to be present in every case of the disease, and it must not be present in healthy individuals. The second requirement is that suspected pathogen, it has to be isolated and then grown in pure culture. And then a healthy test subject, if they're inoculated with the suspected pathogen, they have to develop the same signs and symptoms of the disease. And then finally, that pathogen must be re-isolated from the new host and shown to be identical to the pathogen in postulate 2. So we can see that example here. Here we have they're experimenting with mice in a particular pathogen. So here's the healthy organism not experiencing any of those symptoms, and that healthy organism, we're looking at the blood underneath the microscope and we see the red blood cells, but we don't see the agent, and then we can plate it, and on the plate we don't see any causative agent growing up on this plate that has been made in order to select for that causative agent. On the other hand, here we are meeting requirements 1, 2, 3, and 4. So we take a diseased organism suspected as having the causative agent, showing the signs of that disease, and we are able to acquire that suspected agent from that organism. We're able to isolate it, and then we're able to give that to a healthy individual, and then lo and behold, it causes the disease. And then we can isolate that disease, that causative agent again there. Going through this process, we can see that we've demonstrated that the disease is in fact being caused by an infectious agent. Something I want you to be cognizant of is that while this is a pretty bulletproof way of demonstrating a disease is caused by a particular agent, this is a very high bar in order to meet all these check marks. As you can imagine, not all microbes are culturable. We said, and I think it was the first lecture, almost 99.99% of microbes are not culturable. And in fact, a lot of microbes that cause diseases, they are very selective in what they can grow in. So they're very difficult to grow, and we might not be able to grow them in pure culture. And then this is a little bit of a issue. How do we infect other organisms with that disease? Now here we have mice. It's quasi-ethical to experiment on mice in order to maybe learn some important things that could save humans down the road. But this is really hard to do if you have a pathogen that might only be found in humans, or might not be a causative agent that you can give to mice very easily. And so, as you can see, there's going to be a lot of difficulty in using Koch's postulates in order to demonstrate these diseases are caused in humans, and that sometimes is the case. For example, Lyme disease is really hard to figure out what exactly was causing Lyme disease, and many other diseases out there. So this is still a area of growing interest in people who study diseases. And finally, let's see how we compare the virulence of different organisms. We can do it in two ways. We can look at the infectious dose versus the lethal dose. The infectious dose, abbreviated as ID, is the number of causative agents that it requires to cause the disease. It can be signified with a subscript, and usually we look at the ID 50, and this means the number of organisms required to infect about 50% of the population. So in this example, whatever amount of cells or viruses we used, it caused 50% of the tested animals to become infected. Now, tested animals are not always going to be humans, so sometimes we use them in order to make some assumptions about humans, but not all diseases can you just simply see how many humans it infects. The LD50 is a little bit more intense. That stands for lethal dose. So how many cells or viruses did it require? Or it could even be a toxin, for example, in both of these, ID or LDs, that 50% of the tested animals died as a consequence. So what does this mean? Well, it means that the lower these values are, that indicates a higher level of virulence. Think about it. If it requires fewer cells or fewer viruses to do the same damage than another causative agent, then clearly it's a more virulent disease. So as an example here, the infectious dose 50 for norovirus is only 1 to 10 viruses. It requires very little norovirus to get someone sick, and that's why noroviruses, which are commonly found in cruise ships, you hear stories about norovirus being spread through the cruise ship. It spreads so easily and so quickly. Whereas if you look at the ID50 for enterotoxigenic E. coli, sometimes called ETE, it causes traveler's diarrhea, and it requires a lot more cells, about 10 million to 10 billion cells. I know this might seem like an astronomical amount of cells, but trust me, 10 billion cells is really not that many cells in the microbial world. And that wraps it up for chapter 15, and we're going to now move into chapter 16 and discuss disease and epidemiology. Epidemiology is the medical science that studies the incidence, distribution, and rates of diseases, as well as their possible controls or maintenance of those diseases. Incidence means the rate, the occurrence, and the frequency. And again, we're going to be using these two terminologies here, signs and symptoms. Signs are objective changes. We can measure them, whereas symptoms are feelings. They're subjective. They're the way people feel as a result of the disease. And each disease has a set number of periods that they tend to all follow. This is a very common pattern that we see here. And what we see on these two axes or axi is the red line and the blue line. And it says here the red line represents the number of pathogenic particles. They're saying pathogenic particles because remember, not all pathogens are living. We do have viruses. And so we can see them along the red line here in this kind of bell curve shape, and then the severity of symptoms shown in blue. And we can break up these periods into the incubation period, prodromal period, period of illness, period of decline, and period of convalescence. And depending on the pathogen, a patient may be contagious for only certain periods, or maybe they could be contagious for all 5 periods. For example, the Epstein-Barr virus, called typically abbreviated EBV, it's shed in the saliva of individuals who are carriers of this disease for months following recovery from mononucleosis, which is what it causes. Sometimes you just hear it called mono. So let's take a look at each one of these different periods. The incubation period shown here is the period of the initial infection and appearance of signs and symptoms. This length is highly variable depending on each disease. And we're going to find some of the lowest concentrations of the pathogen, obviously, here. Typically, you have no signs or symptoms here in this period of incubation. And an example of how this can vary so much is if you look at rabies, for example, it is a very short incubation period. Whereas if you look at a disease, say like AIDS caused by HIV, it has a very long incubation period. The prodromal period is typically a short period of time. It's a short interval between the incubation and the period of illness exhibited by some diseases where there might be some mild symptoms that are experienced, very mild though. So you're wondering, maybe am I getting sick? I feel a little funny, but it's not too serious at this point. Prodromal. The period of illness is the time where things really get kicking. So this is the most severe period for a disease. So those signs and symptoms are the strongest during this period. And also you're going to find that the pathogens are in their greatest concentration in this period of illness. And then you have the period of decline. This occurs after the host's immune system is creating a response and overcoming the infection. And signs and symptoms start to subside. The pathogen numbers are declining here. However, we need to be careful during this period of decline. Your system may be susceptible to getting secondary infections due to opportunistic pathogens that may otherwise be benign. So you might have pneumonia that pops up or something like that. Okay, and then finally we have the period of convalescence. And during this time, the body recovers and returns to normal function. Some diseases can leave permanent damage, which we call sequelae or sequela if it's singular. So, for example, people may have heart murmurs following a case of scarlet fever. And sequela is just a medical condition or complication that arises as a result of a previous illness, injury, or medical intervention. All right, so no discussion about disease can be had without some discussion about what is the scientific study that looks at disease. So epidemiology, this studies disease from a population perspective. So it's kind of more big picture. So it might look at a variety of things to cover a population perspective. It might look at the etiology of a disease, which means what is the cause of the disease, and the geographical distribution of the disease, the timing of infections during their disease occurrence, the transmission of the disease, and we can classify disease by how communicable it is. Well, what does that mean? Well, non-communicable means that it cannot be transmitted between hosts. Communicable means it can be transmitted from one host to another. And finally, contagious means it can be transmitted rapidly and easily between hosts. So in epidemiology, we might also look at the occurrence of the disease. And morbidity is the state of just being in a disease state. The morbidity rate is the frequency of occurrence of a popular disease in a population. So this is a little bit different than incidence. Incidence is the number of people who contract a disease during a particular time. So these are only the new infections at a particular time. So a great way to examine this is by looking at human immunodeficiency virus, HIV, and we can look at all of the new contractions each year by looking at this blue line. So that would be the incidence. You could look at the incidence in 2010, in 2005, and 2000, um, and so on and so forth. Now, prevalence is the number of people affected by the disease during a particular time. So it's not just a new case. So incidence should always be lower than prevalence. It's not just a new case, but it's the cumulative number of cases at that time. Now, HIV, there is no cure, so to speak, that is well demonstrated. So these are cumulative. So someone who was part of the incidence of 1995 is going to count if they're still around in 2005, and this would be additive. So the prevalence is the total number of people that have that disease at that time, whereas incidence is just the number of people who gained that disease at that time. Mortality is different than morbidity. It is actually the result of the disease being in death. We can see different mortality rates here depending on diagnosis and treatment. Okay, we can look at the pattern of incidence. Remember, incidences are new cases, and we can break them down as sporadic, so they occur only occasionally in a region. So, for example, plague, something that happens just occasionally. Endemic means that they're always present in a region. So, malaria in sub-Saharan Africa. Epidemic is when it's present in a large number of people in a region. So you might have an Ebola that's epidemic in the DRC. And a pandemic is a global outbreak, not just a region, but globally it is an outbreak of that disease that affects a larger number of people. And we can look here and see normal seasonal variations versus an epidemic status. So a normal seasonal variation is just going to sort of fluctuate as it normally does with the maybe so many years, so on and so forth. And we can see how a epidemic is caused here during this period where this is a big outlier event. We could classify diseases based upon their severity as well. Acute means that it develops rapidly but lasts a short time. An example of this is the flu virus. Chronic means that it develops slowly and is less severe, but it lasts a long time. For example, hepatitis B. Subacute means it develops at a medium rate and to medium severity. So endocarditis is a subacute disease. And latent means that causative agent produces disease after an initial period of inactivity. So, for example, if someone has cold sores, that retrovirus that causes those cold sores, it goes dormant or latent for a period of time, and then it can reemerge later on. We can also examine the extent at which it happens throughout the body. A local infection happens where the pathogen only causes disease at a specific small area of the body. For example, here's toenail fungus. And systemic means that an infection occurs throughout the body. This can typically affect a system, so the circulatory system or the lymphatic system. And we can also look at the reservoirs of infection. So where these diseases can be transmitted from living organism or an inanimate source. We call those sources reservoirs. They can be living or non-living. Living, so it could be humans that are both affected and carriers, or maybe it comes from wild and domestic animals, for example, the plague. And non-living reservoirs could be, you could get it from the soil, so you can get botulism from the soil, or polio from a water sample. We can categorize the different types of contact that diseases can come from. Direct contact transmission is when there's direct physical contact between the hosts. Congenital transmission is where it's passed from mother to fetus, and this occurs at birth, of course. Indirect contact transmission is where there's something in between that carries it from one to the other through a fomite, which is a non-living object that serves as an intermediate. So maybe a doorknob or something like that you touch, and someone else touches that and gets if you have the flu, they pick up the flu from you from that source. And then droplet transmission is different than direct contact, but it's related. So it's from host to host, but it's where the pathogen is spread a short distance through mucus droplets created through coughing, sneezing, talking, etc. So that is the type of contact. And then the vehicle that transmission occurs in can be of many ways. So it can be airborne transmission. This can be transmitted through long distance droplets from dust or very fine spray or respiratory expulsions. It can be waterborne, or it can be foodborne. And then lastly, something called the fecal oral transmission, where it's transmitted from the feces to another vehicle such as food or water, and then to a host. Now, there are two types of vectors that can transmit disease as well. And vectors are animals that carry pathogens from one host to another. The most common vectors for human diseases are actually insects. And insects can do it in two ways. They can be a vector by mechanically transmitting that pathogen. So this is where it moves passively via just contact with the insect body part. So here's this fly. Imagine this fly was somewhere it shouldn't have been. It picked up a disease. It lands on your food at a picnic, and then you eat that food and you get whatever disease it's carrying. That's passively done through contact. That would be just a mechanical transmission. And as you can imagine, biological transmission is where that insect actively causes the infection. So if that insect bites you or penetrates your skin, it can directly inject you with that pathogen, causing you to acquire that disease. And that wraps up our discussion of chapter 15 and 16. Thank you so much for joining in, and we'll see you on the next one.